Today I will be talking about the effects of different levels of salinity concentration on the enzyme trypsin. Enzymes are very important as they help lower activation energy in chemical reactions that take place in cell by deforming the substrate molecule bonds and making it breaking down those large molecules into smaller ones. This is important as activation energy is far too high otherwise. So the enzyme that we use is trypsin. Trypsin is located in the small intestine, the lumen of the small intestine. The ideal conditions for the enzyme is pH 8, varying levels of salinity, and body temperature or slightly warmer than body temperature. Trypsin digests dietary proteins, and we were trying to figure out at what concentration of salinity does trypsin function the best. And our conclusion was from that the salinity of 0.15 molar. And we came to this from our previous experiment where we did 0.3 moles, and from that experiment we figured out but 0.15 would be better, as we saw that 0.3 did not work as well, and other groups that were testing other conditions had the said salinity of 0.15 moles. So the materials and methods. So for the reaction buffer, we did 450 microliters of it at from 0 moles to 0.6 moles at pH 8 and 45 degrees Celsius. Then we added an azocasein, which is the dietary protein that trypsin digests. Then we added the trypsin in, then TCA. What is TCA? TCA is what denatures the trypsin, and then basically stops any enzyme activity. After that, we did water bath at 45 degrees Celsius, which we came to from looking at other groups who did temperature, and that seemed to be the best, as warmer temperatures than room temperature was the best for the enzyme. We centrifuged for a few minutes, and which helped by forming the supernatant of the undigested azocasein and the enzyme, leftover enzymes, and that formed a supernatant. And the liquid, we took out the liquid and we put it in the spectrometer at 440 nano wavelengths, I believe, and our results were inconclusive as we came to negative numbers, which should really not happen, and that happened due to human error as we were rushed. Our teacher gave us the results of another group that helped basically come to any kind of conclusion for our groups. From that we came, but they did have different negative controls. Our negative controls were set by replacing the enzyme with distilled water, with the same volume of distilled water that we would have done for the enzyme. So I'm not sure what they did for their negative control, but I'm sure they did that fine. So from that we took the experimental average and etc., and we came to the null hypothesis. Null hypothesis really helps us figure out if there's a significant difference between the enzymes, and from our experimental design was adequate, but execution was flawed due to human error, but other groups' results did help. So from that, we came to the conclusion from group 1, section 4's data that our thing was somewhat correct, as trypsin's optimal enzymatic activity is at 0.75 molars to 0.3 molar, and 0.15 is between them. The p-value at those numbers were not significantly different. Our control variables were the temperature and the pH. The pH was already set in the reaction buffer, and the temperature we came to by seeing that the temperature, the warmer temperature would be better. 40 to 50 degrees seemed the best, as our teacher had pointed out, and our group decided at 45 as that would be the middle. From the difference between the first and the second experiment is that the amount of replicas we had, we significantly increased our replicas for all the different levels of concentration of salinity. We had 4 replicas and 3 replicas at for the negative controls. And from that we learned that more replicas are better, and we also learned new things like null hypothesis and etc. From this, I came to the conclusion for the ideal conditions to grow bacteria are lower levels of salinity, as our experiment shows that lower levels of salinity show the best. thrive for the enzyme. pH higher than 7, as we saw from other groups, with their pH ranged, the best activity ranged from 7 to 9, I believe, and warmer temperatures. Our references, one that I would really like to point out is Group 1, Section 4, Spring 2020, as we use their data to come to any kind of conclusion, or basically do our whole experiment as our data was incorrect. Well, thank you.